All right, so I think that's about it. Excellent. So good morning, good afternoon, good evening. Welcome to this week's edition of Paleo Perks. My name is Elizabeth Seibert, and I'm one of the Paleo Perks committee members. And today it is my great pleasure to welcome Jihad Rachid from the Hassan II University of Casablanca in Morocco. Jihad will be giving a talk titled Tracking Climate Variability and Environment Changes of Carbonate Mounds and Sediments in the Western Mediterranean over the last 14,000 years. So for those of you who haven't been to a Paleo Park seminar before, I just want to give you a quick expectation of what to expect over the next about hour or so. So we're doing a quick welcome and announcements. Jihad will then talk for about 30 minutes, followed by a moderated Q&A. After the formal part of the seminar, we invite you to stick around for an after-talk tea time with our speaker. And we'd like to remind you to please send any questions that you have or technical issues via the chat function to the Paleo Perks questions host, who today is Pedro. So just a little bit of quick housekeeping. Paleo Perks values the participation of all folks interested in paleo sciences, and so we ask that you please abide by our code of conduct during today's seminar. If you've somehow made it into the seminar without signing up to the email list and reading the code of conduct, please make sure you do that at some point today. We like to ask you to remain muted for the duration of the talk. You should not be able to unmute yourself. However, if you find yourself unmuted at any point, please go ahead and remute yourself. You can ask questions by chatting with the questions at Paleo Perks host, or you can actually use the little raise hand function, which I believe is currently down in your reactions tab, and we'll let you unmute and ask your question. Any technical issues should also go to Pedro today as questions at Paleo Perks. If you would like closed captioning on today's seminar, we're using the built-in captions with Zoom, so you can turn the CC button to show or hide them. And as a note, if you would like a different kind of captions, please reach out. We're always willing to work with you. We'd also like to remind the community to nominate outstanding early career researchers to give Paleo Perks talks. We're always looking for up-and-coming folks in the late stages of grad school and postdocs, and we'd love to be able to feature you. And finally, we'll be dropping in the chat or a weekly demographic form. If you feel like letting us know who you are, that gives us an idea of who's coming to these seminars and a little bit of feedback. All right, so enough talking. Today's speaker is Jihad Rachid. Jihad did her undergraduate in Mohammed V University in Rabat, Morocco, and then a master's at Hassan II University of Casablanca, also in Morocco, and is now a PhD student working on environmental geology and paleopalynology, also in Casablanca. And so I'm really excited to hear what she has to say, and microfossils are my favorite. So now it's all to turn it over to Jihad. So have a great talk, and I can't wait to hear about the microfossils.
Thank you so much. Okay, so just as a test, do you see the slide?
It looks perfect. Have a great day.
Yeah. Okay, yeah, all right, then I will go ahead. Good afternoon, everyone. My name is Jihad Rachid, PhD student and teaching assistant at the Faculty of Sciences and Hassan II University of Casablanca, Morocco. Well, firstly, I would like to say thank you so much for inviting me, and thanks to all the organizers, I mean, for putting such a great series of all paleontological studies. And yeah, it's really amazing and an honor as a PhD student to be among all these researchers from all around the world. Well, so thank you so much again. Well, so today I'm going to be talking about the first paleontological study on carbonate mounds of cold water corals in the North Atlantic and Western Mediterranean Sea, and exactly over the last 14,000 years. Well, so this research focuses on the qualitative and quantitative study of the biotic content dinoflagellistes, pollen, plankton foraminifera in the sediments of carbonate mounds in the North Atlantic and Western Mediterranean Sea. Well, the study examples, I mean, of my research thesis, and even for this present talk, is are all book scores and on-mount gravity scores that were collected during an oceanographic cruise, which were performed in 2013. Well, the general purpose of this entire mission was to collect a variety of cores, I mean, from calypso core, gravity cores, box cores, for different studies like paleoceanographic, biogeochemical studies, even for micro and macro paleontological analysis. But the main objectives of my present work is about paleo-environmental and climatic reconstruction based on dinoflagellate cysts and pollen during our study interval, which is Upper Pleistocene to Holocene. And our second objective is detailed assessment of the impact of environmental and climate variability on the development of these cold water corals carbonate mounds in the North Atlantic and Western Mediterranean Sea. Well, in this talk, I will try to present for you two different contributions. The first one will be focused on book scores. So the study, I mean, on Moroccan Atlantic margin in the northwest of Atlantic and exactly in the Gulf Cadiz. And the second part of our first contribution, it will be the Alban Sea. Which is, as you know, the most western part of the Mediterranean Sea. Here, this map will show you the geographical location of our book scores. Actually, we have 5 book scores. We have 1 book score in the Moroccan Atlantic margin with a bathymetry of 503 meters. 503 meters, and we have 4 different book scores in the Moroccan Mediterranean margin and exactly in the Alban Sea with a bathymetry, as you see, between 320 meters and 470 meters. Here, I just want to show you some pictures I took it during the research oceanography cruise. And here it is the book score I used and our 5 studied book scores. So actually, when we talk about book scores, they are really short cores, which dim length doesn't exceed 1 meter. And for our studies, 5 book scores, their dim length is between 40 centimeters and 1 meter. Well, after we took our sediments, during our research oceanography cruise, we took our sediments to laboratory for chemical treatment. So firstly, we attack our sediments with hydrochloric acid to eliminate carbonates, and then we attack our sediments by hydrofluoric acid to eliminate silica minerals. Then we save our sediments under 10 micrometer to eliminate clays. Then we get our target, which is organic residues, which will be mounted after on slides for subsequent observation under microscope. Well, for our microscope analysis, we found that all the organic residues for all the studied samples of all the 5 studied book score are really rich and diversified on different polymorphs. And here I'm showing you some pictures of our real material. And these pictures are from our first contribution and second contribution. I will show you later about the second contribution. Well, so we found different polymorphs. We found foraminifera, pollen, and also a great dominance of dinoflagellate, which play a really great tool for construction of the paleo environment. Yeah, so here I'm just showing you some dinoflagellate and some pollen recorded in all our studied samples. So now for the results of our first contribution, as I told you, we have just 1 book score in the Moroccan Atlantic margin. And as you see, it is book score of 90 centimeter of sediments with different lithologies from muddy migrate to migrated mud with different colors, as you see in the legend, brownish, grayish, etc. And their age span between 12.09 and 6.8, means we have from the Upper Pleistocene to the Middle Holocene. Well, they're cold water corals. Well, our first objective is reconstructing the paleo environment. So first we will try to see what was the environment in the past. So what we will do, we will be based on dinoflagellate associations. So we will begin by the base of this book score, which is the Upper Pleistocene, and which is characterized by high percentage of cold taxa like nematodes, Phyreopsis labyrinthus, Impangidinium sphaecium, Spinifarius largus. These cold taxa indicate that we have, that we have an alternative to oceanic environment. And as you see, upward the Holocene, you see that these cold taxa will significantly decrease, and even some of them will be absent after, because they will be replaced by other species, which are the warm taxa. And then we will go from an alternative to oceanic environment to an environment, sorry, the slides go away from me. Yeah, anyway, so we have an outer to internetic environment during the lower Holocene. And then, as you see, we have a maximum extension of this warm taxa, which is the Spinifex, mirabilis, and Impagidinium aculatum, which indicate that we have warmer waters and also indicate that we have internetic to oceanic environment. Also, we have high percentage of Languidinium maculatum, and this species indicate that we are in an environment really rich in nutrients. Means that all the sediments of all this book score were deposited in environment really rich in nutrients, possibly a fall from the seafloor. Yeah, so that's for the environment. Now for the climate. So after the paleogeologic assessment, we found that we have high percentage of the marine fraction, which is presented by basal foraminifera and dinoflagellate cysts, compared to the continental fraction, which is presented by pollen. And even we have this low percentage of pollen, we could reconstruct the climate. So what we do, so we'll do the same as we did for the environment. We'll begin by the base of this book score. And as you see, we found high percentage of folded taxa, but exactly we found high percentage of altitudinal conifers coupled by tree and shrubs pollen. And after will be followed by high percentage of herbaceous pollen, which indicate that during the lower Holocene, we had arid climate. And at the top of this book scores, during the middle Holocene, we had high percentage of tree and shrubs pollen, which indicate that we have humid climate. So now for the Moroccan Mediterranean margin, as I told you, I have 4 book scores, but because we don't have plenty of time, I will try to show you just one example. So as you see, this book score is a book score of 80 centimeter of sediments. So we have grayish muddy micrite from the base to to 15 centimeter, and then we have a brownish micrite mud. From 50 centimeter to the top of the book score. And as you see, we have only middle Holocene sediment characterized by high percentage of warm taxa. And also, again, we have high percentage of Languidinium maculatum, means we have internetic to oceanic environment with an environment which is really rich in nutrients because we have Languidinium maculatum. Now for the climate, at this time, we'll begin by the top of this book score. So here we found high percentage of temperate pollen, but exactly we found high percentage of herbaceous pollen. And another thing, this ratio of heterotrophs, autotrophs can play a really great role as a high temperature proxy because we will have a lower value indicate that we have warmer waters. Now at 6.29, we find high percentage of tree and shrubs pollen, which indicate that we have semi-humid climate. Yeah, here, just to sum up our first contribution. So here, as you see that I presented here all our phases for the 5 book scores. And I presented the dinoflagellate associations and each association, which environment indicates, and pollen associations and each association which indicate as a climate. But I added new information. Well, one of the objectives of the research oceanographic was to understand why we have this dispersed cold water corals. So we were trying to create a link between our paleontological results and see if we can reveal anything related to this dispersed cold water corals. So they found the groups who were working, on the cold water corals. They found that the Pleistocene, the upper Pleistocene was a phase which were characterized by high percentage of cold water corals. So we had really good, all the favorable conditions for the proliferation of corals. And it was the case for our results that we found depends on corals, we found a cold climate. But during all the Holocene, you see, we had this cyclicity of climate, of arid, humid, semi-humid climate, and even warm, dry climate. But you see that we found disappearance of corals, but with the appearance of new species, means that we didn't find a total absence of cold water corals, but it was the appearance of new species. So we could deduce that the continental climate change doesn't have significant impact on the demise or the growth, but maybe hydrodynamic conditions, maybe nutrient supply, or maybe even taxonomy can play a significant role on these features. Well, our results were published in a review of paleobotany and palynology in 2020. And now for our second contribution, we work at this time on the samples collected from the gravity cores in the Alboran Sea and exactly in the Western Minya province. So example of the MD51. So when we talk about gravity core, means we talk about a long core. And it is the case for our study. We have this core is of 5 meters of sediments. And as you see, only migrated mud with different colors from the base to the top of this box core. And it is span between 13 to 79 to 4.51. And means we have from the upper Pleistocene to the upper Holocene. So we have, again, the same objective. We try to reconstruct the Kali environment. So we'll try first by saying what is the environment in this gravity core. So in the base of this box core, we found that we have inner neotic environment because, as you see, we have high percentage of heterotroph species compared to autotroph species, and especially the group of Bricantidinium. And this indicates that we have inner neotic environment, and this environment will remains inner neotic until these around this sample of 411 centimeter, where you can see that these species will decrease significantly, and you can see that we have high percentage of cold water, cold taxa of dinoflagellates, and this indicates that we have outer neotic to oceanic environment. And this coincided with the cold event of the Younger Dryas. Yeah, and after, as you see, after the Holocene, you see that these autotrophs will decrease and will be replaced by the autotrophs, which is increasing. And as you see, we have high percentage of 2 warm taxa, Impagidinium aculatum and Spiniphytus mirabilis. So we will have inner to outer neotic around 11.24 and 49, and we will have inner neotic to oceanic environment around 9.56. And after, as you see here, from the lower the phase between the lower Holocene to the upper Holocene, and also during the upper Holocene, you see these 2 warm taxa will show really high percentage, and then with a maximum extension during the upper Holocene. And this indicates that we are in inner to oceanic environment. Also here, you see that we have high percentage of Languidinium archaeoform along all this core. This indicates also in the Alboran Sea, our sediments were deposited in environment really rich in nutrients. Yeah, so now for the PCA, as you see, so the PCA here revealed that the PC1, and the PC2, out of 32 samples, and we have 21 species for this gravity core, explain like 58% and 13% of variance respectively, and the orange dots here represent the autotroph species, and the green dots. So as you see, the presence of this heterotroph species along the PC2 can suggest that the PC2 represents the productivity, and the main compound of PC1 reflecting the temperature in nutrient-rich environment, because as you see, we have Languidinium archaeoform. Well, so we have the first group coincided with the base of the gravity force. And we have the second group, which is characterized by a cold taxa, coincided with the cold, the cold event of younger dryas. And the third group is which is characterized by high percentage of alternating species, coincided with the lower Holocene. And then we have the fourth group, which coincided with, the phase between lower Holocene to upper Holocene, and also the upper the upper Holocene, sorry. Yeah, so now to reconstruct the climate. So we have, again, as you see, I mentioned in the figure caption, we mentioned exactly the species for each group, of the tree and shrubs, and also for herbaceous steps, elements. So we will do as we did for the environment. We'll begin by the base of this book score, and you see that we have high percentage of tree and shrubs colon, and this indicates that during the boiling Alurod we had a warm and humid climate, and this climate remains warm and humid until around the sample 411 centimeters. At this moment, we'll have high percentage of altitudinal conifers and high percentage of herbaceous pollen, which will indicate that we have cold and dry climates. Then after, as you see, we have high percentage of herbaceous pollen, and this indicates that we have warm and dry climate along, the lower Holocene. And then we will have warm, humid climate, at around 9556, and will remain warm and humid until the upper Pleistocene, where we will have high percentage, as you see, of herbaceous pollen. Yeah, so now just to conclude and to sum up our second contribution, I try to, present again, we try to create a link between our paleontological results, and we will try to see if we can reveal again something about the cold water corals and the mice. So as you see, I presented here all the phases with the results of the group who they are working on cold water corals. And I mentioned here all, each type of environment based on dinoflagellates. So as you see, during the upper Pleistocene here, which is presented by the boiling Alurod and the phase between boiling Alurod and younger Dryas, you see that the environment is really rich in reduced, the environment known by reduced salinity and also characterized by lower oxygen content and rich in nutrients. And we found high percentage between 30 to 70%. So we can, we can explain that despite this, that the bottom water being depleted in oxygen content, the cold water corals, as you see, they were able to proliferate, recording really high, the highest, percentage between 30 to 70%. But compared to the younger Dryas and Holocene, you see that we have a total, absence of the cold water corals, or a gradual decrease, which take from 50 to less than 5%. Even you see that we have all the favorable, favorable conditions to the proliferation. We have well-ventilated waters. We have waters which are really rich in nutrients. So the only thing that we can deduce that not only temperature, as we see in the first contribution, or the oxygen content can play a role on the mice or the recruits. But anyway, I can't reveal other parameters. We found many another parameter which can play a really significant role on the mice. But our publication now is under review in marine micropaleontology, and I hope that I can meet with you again and reveal all the results of this second contribution. And we'll be happy to show again, as future work will be this time working on the northwest of Atlantic. As you see that like 90% of our work was based only on the Albertan Sea, but this time we will work on the northwest of Atlantic. So I hope that you enjoyed my talk, and thank you so much for your attention. And yeah, I will end here, and will be happy to hear all your questions and discuss with you. Thank you.
Excellent. Thank you so much for that. That was really, really neat to see all of the microfossil work you've done and all of the palynology, really. So we have a couple of questions coming in, and I'd just like to remind folks that you can send questions in the chat, either to the chat to everyone like we have here, or to the questions at PaleoPerk's host, who is Pedro, and we can read those out anonymously for you. Or if you would really like to ask a question verbally, you can raise your hand and we'll work on that as well. So actually, we can start with this question from Elsie in the chat. It says, firstly, I would like to thank you for your wonderful talk, and I would like to ask you, could I use dinoflagellate cysts on sediments of landslides?
Well, yeah, actually, when we talk, well, dinoflagellate cysts are really good microfossils for paleoenvironmental reconstruction. But when we talk about landslides, so the sediments are enough mixed up. So it's, I mean, based on dinoflagellate cysts, because the sediments are already mixed up. So even the microfossils will be mixed up. So you cannot know. They can come from everywhere, and they can anytime. So I think it's not a good idea to use dinoflagellate. Or even other microfossils, not only dinoflagellate.
So I'm also going to follow this next question, which I will also put in the chat. It says, where is the pollen coming from for each of these box cores? There's a lot of different land masses and environments around that area of the Mediterranean. Could some of the changes that you're seeing be due to changes in source material rather than changes in plant composition of that same location? Particularly, I think maybe due to with the pollen records.
Okay, I'm trying to read again the question. And if I want the material, could some days change be changing source material location rather than changing plants composition and distant location? Well, actually, we found that all the pollen and algorithm, as you see for the box cores, we were working, in the Moroccan Mediterranean margin. So most of the pollen were coming from Morocco. And we found some pollen coming from the Spain and Portuguese for the Northwest Atlantic. I hope I replied on your questions, if it is that's what you mean.
I think that gets it, at least some of that. And we can clarify as well if we need to. I think one other question we have here, which I'm also going to put in, and then we can get to Elsie's follow-up there. It says, how do records from other microfossils compare, e.g. nanoplankton or foraminifera?
How do records from other microfossils compare? What do you mean? Do you mean our construction environment? How do?
I think so. Like, do you see similar changes at the same times in nanofossils or foraminifera? Has anybody looked at the other microfossils?
Yeah, there is others people, they work it on foraminifera, and we could, we had the same results, and others we couldn't find the same results, because dinoflagellate can give you correct environment, and can give you also the indication about the climate. And we use pollen for, correct answer about the climate. So there is a group who worked on the same course by foraminifera. We had same, sometimes we could, agree, in others we couldn't agree, because dinoflagellates can't lie.
I think once, because they all have slightly different records. I think that's something that I've always found really interesting, looking at all of the deep sea sediment, like IODP cores, where you have, 5 different ages from 5 different microfossil groups.
Yeah, you're right.
So I think I'll ask this question from Elsie, asking, what about coarse sediments? Can we work on coarse sediments for palynological studies?
I think she's working on landslide sediments. Actually, we found, sediments, great abundance of dinoflagellates in fine sediments. And when you will work on coarse sediments, you will have a lesser chance to find dinoflagellates, or even you cannot find any dinoflagellates. If you read many articles about dinoflagellates, you will see that people working on really fine sediments, as my case. So I don't advise her to work on landslides, sediments. I think it is the same person, yeah.
Yeah. Also, if you want to stick around afterwards, we can chat about methodology during tea time, and maybe that will answer some more questions. I'm just going to move on to our next question, which is, how does diagenesis affect dinoflagellate cysts?
How does diagenesis affect dinoflagellate cysts? Well, actually, I didn't face a case about diagenesis, which can affect dinoflagellate cysts. So if I said something, I would say all the wrong answers. I never had the chance to read anything about diagenesis with dinoflagellate cysts, so I will be searching about this. Which is a great question.
Excellent. So one more question here. It says, in modern marine ecosystems, there can be big shifts in the ratio of autotrophic and heterotrophic plankton that record changes in nutrient availability. Do you think that you are seeing some of this signal in your box cores?
Well, actually, there is no confusion between the first contribution or the second contribution. I think I got heard, or here's the question, because in the first contribution, we're working on really short box scores. So in the short box score, it was really hard for us to see exactly these fluctuations of the environment and the climate. So for our first contribution, we said, yes, the climate doesn't, affect, or has no significant impact on the demise. But in our second contribution, we worked on really long reports, so we could see exactly this fluctuation of environment climate, and we could know exactly that, yes, temperature play a great role on their demise, but it's not the only parameter. And I said that I hope that I can be here again and show you what the parameter which we found it. Sorry, because I can't reveal all the results till it's under review. But hopefully we can meet again and can give you all the results. I think there's another question in modern marine.
Yeah, there's one more question that I have that I just put in the chat, which I can read again if you would like.
Yeah. You think you are saying some of the similar in your book scores. Yeah. Sometimes I found like confusion about, this ratio of heterotrophs, autotrophs. But sometimes I said, because I was comparing the results of pollen and dinoflagellate cyst. And if you read the article of Pispalova, 2008, you will see that she had the same. Sometimes it's match perfectly her results, and sometimes no. And the first time I used this proxy was great for the box score in some phases, but I just know. Even if you will see some, even dinoflagellate cysts, but you cannot, focus it more on this ratio. It can help you, but you should not focus it more on it.
Excellent. Well, thank you very much. I'd just like to give anyone another 30 seconds or so to add any last questions that they'd like to have. And if not, we will go ahead and maybe I will get that. Excellent. All right. So promise I can do Zoom. So I think we'll go ahead and wrap up there. So thank you so much, Jihad, for your really interesting talk, and thank you to everyone else for joining us today. I just want to go ahead and wrap this up. We'll say we'll drop this link to our weekly feedback in the chat. And then we'd like to invite you to join us next week when Brian Shirley of Friedrich Alexander University in Nuremberg, Germany, gives a talk titled Big Equipment, Tiny Structures, a Multi-Analytical Approach to Unraveling the Conundrum. But if you have not gotten enough paleo for your day, we'd like to invite you to stick around for the next about 20 to 30 minutes to have a nice tea time chat with Jihad. And she can tell you a little bit more about her career path and the work she's doing. You all are welcome to come hang out with us and join us for a little bit of paleo fun. So we'll go ahead and give us about a 2-minute break, and when we come back, we'll have our tea time. So thank you again for your really interesting talk. 2 minutes.
Thank you.